Is 2020 spring semester. We are working on the next unit, Unit 6 Microbial Genetics. So far, I haven't done an audio for Unit 5 Microbial Growth. The reason is we're now in the midst of the novel coronavirus outbreak, and we're not sure if schools will remain open. So my thought was I will try to make audios first for the lectures and labs that we're currently in case we have to cancel classes, and I would hope it would only be for a week or so that we'll have a bank of videos so that we can stay on track in both lecture and lab. Okay, so this is going to be our microbial genetics, our first PowerPoint. So in this first PowerPoint, we're going to be describing DNA replication. We'll be then describing how the cell uses the information in the DNA sequence to transcribe the DNA into an RNA sequence, and then use the messenger RNA sequence as a guide to make proteins. So thus the three topics are DNA replication, transcription, and translation. This is an important concept map, worldview of information flow within cells, and this is called the central dogma of information flow in cells, developed by Francis Crick of the famous Watson and Crick duo that described DNA structure. So we need some vocabulary, so I'm going to be using the term template, T-E-M-P-L-A-T-E, and a template is a guide. So when we're describing information flow, we always ask, what is the template and what is being made? And we want to recognize that these processes will be carried out by DNA polymerases and RNA polymerases when we're making copies of DNA or making RNA. So in this first example, in this cartoon, we have an arrow, and the origin of the arrow starts with DNA. So this is telling us that DNA is going to be the template, the guide, and then the arrow loops around and points right back to DNA. So this is our little cartoon for the process called DNA replication. So before cells divide, they have to copy their DNA, because when they split in two, each of the so-called daughter cells have to receive a copy of the chromosome. So our focus will be in bacteria. Most of the early microbial genetics was done in E. coli, so we'll be using E. coli as our kind of our model organism for DNA replication. And then in an entirely different process, the big name could be gene expression. This is a process by which the DNA base sequence acts as a guide to make RNA, and this process is called transcription. And then the base sequence in a specific RNA called messenger RNA or mRNA will be used as a guide by ribosomes to make a protein. And you can see that we're switching from nucleic acid language, the alphabet of nitrogenous bases. Now we're switching to protein language, and we could say the alphabet are amino acids. So since we're switching languages, this process is called translation. Now, this is the flow of information in cells, but we always want to remember that viruses love to break all of our rules. So when we get to the virus unit, we'll see that there's some viruses, they're RNA viruses, they're called retroviruses for backward viruses, the most famous one being HIV. HIV has an RNA genome, and when it gets into our cells, HIV has a special enzyme called HIV reverse transcriptase that uses its HIV RNA as a guide to make DNA. So this process is called reverse transcription. The bad news is once that DNA copy is made, then the HIV inserts its DNA copy into our chromosome, and once it gets in there, we can't get it out again. So that's a process we'll come back to when we do our unit on viruses. So in describing DNA replication, we have to review the structure really quickly. So it wasn't until 1953 that the structure of DNA was described, and it was described as this beautiful double helix. And the folks that wrote the description were James Watson and Francis Crick. They had help. So there was a team of folks that helped with a description of DNA. And this team received the Nobel Prize in 1962. So Watson Crick, and then another scientist, Maurice Wilkins, also received the Nobel Prize. And he and his lab were important because it was in his lab that Rosalind Franklin worked, and she was the one that did the X-ray crystallography that gave the images to Watson and Crick. So they were able to come up with the concept of a double helical DNA molecule. But it was sad. She died before the Nobel Prizes were awarded, and they have a rule that if you're dead, you can't receive the Nobel Prize. But her work was really important. So here's just our cartoon of this beautiful double helical structure, almost like a spiral staircase. And again, we said we're going to focus on E. coli, where most of the early microbial genetics work was done. And just to kind of give us an idea of all the challenges E. coli faces when it's trying to copy its chromosome before it splits into two. This is an electron micrograph. Here's a single E. coli cell in the middle, the little guy that looks like a sausage. And then it's been broken open, and it's released this huge tangle of chromosomal DNA. And it's hard to believe, but this is a single circular chromosome. And what's unbelievable is that the E. coli can package the DNA into this little tiny cell, and furthermore, that it can make a copy of the chromosomal DNA, in this little tiny cell. So we're going to see that the E. coli has a lot of challenges that that it has solved. It solved these problems, found strategies for every step of the way of DNA replication. This is just to give you an idea, another cartoon of the circular chromosome of a bacterium. And you don't need to know what the colors mean, but one thing I thought was fascinating is through DNA sequencing, they know where specific genes are, and this is our key. And to me, what's really interesting is if we look at, I guess, violet color, all of these violet colors are locations of genes that are involved in DNA replication and repair, and you can see just how many there are. So again, a quick review of DNA structure. So DNA are polymers made up of nucleotides, and specifically these nucleotides, we would describe them as residues of nucleoside monophosphates. But we're going to see that to synthesize, to make DNA, the cell has to have reserves of what are nucleoside triphosphates. Those are the energized precursors that the cell needs to actually make DNA. And just again, a quick review, you guys remember that in the sugar, the deoxyribose here, the carbons are numbered 1', and this is to tell us that these are carbons in the sugar. In contrast, the carbons in the nitrogenous base would have just regular numbers, carbon 1, carbon 2. The nitrogenous base is covalently linked to carbon 1' on our sugar, our deoxyribose. Another two more carbons on the sugar that are important is here carbon 5'. We'll see that that's where the phosphate is attached. And then carbon 3' down here, this hydroxyl group is important. DNA polymerase can only form a covalent bond between the oxygen here and the incoming phosphate group of an incoming nucleotide. And we'll see later, because of this, we describe DNA synthesis as being 5' to 3'. We'll use an arrow originating at 5 and then pointing to 3. And that's telling us that incoming nucleotides are going to be added to the 3' hydroxyl end of the DNA strand. The forces keeping the two strands of DNA together are complementary hydrogen bonds. So you want to recall the complementary base pairing rules in DNA is that adenine forms 2 hydrogen bonds with thymine, and cytosine will form 3 hydrogen bonds with guanine. So if I asked you what permits the precise replication of DNA, you're going to tell me it's the complementary base pairing rules. So again, just kind of key points about DNA structure. Again, they're polymers of nucleotides. When we talk about DNA sequence, we're talking about the nitrogenous base sequence, and we always start at the 5' end. So the base sequence on this strand would be TGCA. If we know the base sequence on one strand, we can predict the base sequence on the opposite, the complementary strand. And again, folks, on this opposite strand, we always start at the 5' end. So the base sequence on the opposite strand would be TGCA. It's the complementary base pairing rules, again, that lets us predict what the complementary strand would be. Also notice, folks, that in the double-stranded DNA, the arrangement of the two strands is what we call anti-parallel. That is, a 5' head of one strand is adjacent to 3' tail of the other one, and likewise down here. So that's called anti-parallel. If I were to ask you what links the nucleotides together, they're phosphodiester bonds. It's actually a bridge. So here's one ester bond, there's another ester bond, and then there's our phosphorus in there. So it's a bridge, this phosphodiester bond that links the nucleotides together. As we said, it's helical, often described as a double helix. We would describe how the strands are held together by hydrogen bonds, and the strands are anti-parallel. So again, folks, just want to emphasize the precise replication of DNA is possible through complementary base pairing rules. And here we're starting to take a look at events when, say, the E. coli chromosome is replicated. So this would represent the old parent chromosome, so the two parental strands. And you can see what will happen when the E. coli wants to copy the chromosome prior to dividing in two. It's going to separate the two parent strands. Here's one parent strand, there's the other one. And then each parent strand will act as a guide for synthesis of a new complementary strand. So here we can see the new strand of DNA that's being synthesized using this parent strand. And over here, the same thing's happening. So this opposite parent strand is acting as a template for synthesis of new DNA here. So this type of DNA synthesis, it's called semi-conservative, because when we go from one chromosome eventually to two chromosomes, so here's one new chromosome, here's a second new chromosome, you can see in each of the new chromosomes one old parent strand has been saved or conserved. And again, we have the second strand will be new DNA. So again, when the chromosome has been replicated, we go from one chromosome to two. The new chromosomes will have one old parent strand will be saved, and then the second strand will be the newly synthesized complementary DNA strand. Another thing we want to remember is that DNA synthesis is always going to be 5' to 3'. Okay, 5'. This would be, if we cut it here, this would be the nucleotide that has the free 5' phosphate, and then if we watch the synthesis continuing, this would end up being the 3' hydroxyl end here. And likewise over here, this is, I know this is awkward right now, but on this strand, this template strand, this would be the start of the 5' end of the newly synthesized DNA. And another thing we have to remember is that the new strands are synthesized not only 5' to 3', but they're synthesized anti-parallel to the template strand. So remember, the 5' head of one strand has to be adjacent to the 3' hydroxyl tail of the other one. So this is going to get really complicated when we see how the E. coli is going to copy its chromosome. How can it obey all these rules? So again, this is the, if I ask you what is semi-conservative DNA replication or semi-conservative chromosome replication, you want to remember one old parent strand acts as the template or the guide for synthesis of a new daughter strand. Therefore, each of the new chromosomes will consist of one old parent strand and one new, we sometimes call them daughter strands. Here's the cartoon of the nucleosides. Remember, the nucleoside is a sugar nitrogenous base, nucleoside triphosphates, and these are the energized precursors the cell has to have. The reason the cell needs these energized precursors is DNA synthesis. It's biosynthesis, anabolism, and the cell needs a source of energy to drive biosynthesis. So during DNA synthesis, these hydrogen bonds, represented by the tilde sign, when they're hydrolyzed, energy will be released, and the cell will use some of that released energy to drive DNA synthesis. And we'll see not only will this bond be hydrolyzed, that will be hydrolyzed first, but then consequently this second bond will be hydrolyzed, and that will provide more energy for DNA synthesis. This is just a cartoon, and apologies folks, this figure is from our previous micro textbook, so I need to clean these up, but this is to give you a little bit more idea of the process of DNA replication. So here's the old parent strand right here. And notice orientation, this is the 3' end, this would be the 5' phosphate end. And this is the new strand that's being synthesized. So notice the new strand is being synthesized anti-parallel. The 5' end is adjacent to the 3' hydroxyl end of the template. And then also notice, folks, that it's being synthesized in a 5' to 3' direction. And the reason for this is that DNA polymerase, the enzyme that makes the copy of the DNA, the enzyme that makes DNA, it can only catalyze an electron rearrangement between electrons on the oxygen of this free 3' hydroxyl group. The electrons are going to be rearranged, and we're going to have what's called a nucleophilic, a nucleus-loving attack of those electrons on the nucleus of the innermost phosphorus group. So they call it a nucleophilic attack. We'll have electrons rearranged. We'll have a brand new covalent bond formed between this oxygen and the phosphorus of the innermost phosphate group. Okay, so nucleophilic attack. So this is where the new covalent bond will be formed, right where that broad red arrow is. And then, as you recall, we said that initially this so-called high-energy bond will be hydrolyzed. So we're going to get release of what's called pyrophosphate, and then another enzyme will cleave that second so-called high-energy bond, releasing more energy. And that's the energy that will be used to drive DNA synthesis. And again, the enzyme that carries out this process is called DNA polymerase. And here we can just see, if we take a look here, this is the incoming nucleotide. And here we see that new covalent bond has been formed. There's a new covalent bond that DNA polymerase formed. Now this nucleotide becomes the 3' hydroxyl end. So DNA polymerase will add another nucleotide right there. And the DNA is selecting the nucleotides based on complementary base pairing rules. So again, it's the old parent strand, the template, that acts as the guide to direct DNA polymerase which incoming nucleotide with which specific nitrogenous base should be added here. So we're going to pretend that we have a really powerful electron microscope, and that we're actually watching E. coli as it's copying its chromosome. So just so you don't get confused here, this is the double-stranded circular chromosome of E. coli. It's not the nucleus. Okay, so we're going to see how E. coli will copy its chromosome. So DNA synthesis always starts at a specific place on the chromosome where proteins and enzymes will bind. This is called the origin of replication, sometimes just abbreviated as ORI. So if we'd watch at the start of chromosome replication, it would appear as if a bubble has appeared in the double-stranded DNA. And this is indeed called the replication bubble. So here's an example of the replication bubble. And on either side of the bubble are what are called replication forks. So there's one here and there's one there. It's at the replication forks that all of the enzymes and proteins that are involved in DNA synthesis are madly working. If we watched over time, it would appear as if the replication forks are moving in opposite directions. So this replication fork would appear to move in this direction counterclockwise. This replication fork would appear to move clockwise. So this is telling us that DNA synthesis is occurring in both directions. That's going to make it really efficient. And that's called bidirectional, two-direction DNA synthesis. If we continued to watch, we'd see the replication forks moving along the chromosome until they meet at a place called the termination of replication, or TER, T-E-R. And at this point, we've gone from one chromosome to two chromosomes. So the chromosome replication, the DNA replication is complete, and our two chromosomes will separate. And it's at this time then that the E. coli can divide in two. And then each of the daughter cells, as we see up here, each of the daughter cells will then receive a copy of the chromosome. And then what we're going to do is we're going to zoom in, and this is going to be a little bit harder just on the audio screenshot. Even though it is messy writing it on the board, I like writing on the board because we can take it slowly, like one step at a time. But we'll just go ahead and use the cartoon that we'll have in your DNA replication packet. There'll be some diagrams in there, which the diagram in your packet is actually better than the diagram I'll show you. But one of the problems we're going to try to address, one of the problems encountered by E. coli, is you'll recall, if I back up here, let me see if I can back up to the photo that was showing us all the chromosomal DNA that was packed in. Okay, so if you remember this slide, slide 4, you can see that there's so much DNA packed into that little E. coli. So we might wonder how the heck, how will the cell accomplish that? So the key here is a family of enzymes called topoisomerases. And topoisomerases are responsible for a process called DNA supercoiling. This is when the big circular chromosome is coiled tightly, so it can pack into this little tiny E. coli. One of the kind of parallels that I've heard used is pretend you have lots and lots of clothes to fit into a little tiny piece of luggage. So if you roll your clothes really, really tightly, right, you can pack more clothes into your little tiny suitcase. So we can think of that's what the bacteria are doing, is they're trying to roll or coil the chromosome really, really tightly so it can fit into this little tiny bacterial piece of luggage, I guess we could call it. Well, this creates another problem, though, because if you're going to copy your chromosome, remember you have to you'll recall that we said that the two strands of DNA are going to be separated. The two old parental strands will be separated to create the templates. But if the chromosomal DNA is supercoiled, you can't separate the two strands of parent DNA. It's coiled so tightly you can't break the hydrogen bonds to make the single strand DNA templates. So let me see here. So the family of enzymes that help supercoil and then release the supercoiling, the big family of enzymes are called topoisomerases. And a specific one I'd like you to know for our lecture exam 2 is called bacterial gyrase. And we're going to say that bacterial gyrase plays an important role in relieving the supercoiling so that the chromosomal DNA can be replicated. And we'll also see later, folks, that we have to have release or relieving of the supercoiling for transcription to occur, which is the first step in protein synthesis. Okay, so the reason we're interested in bacterial gyrase, it's a unique target for our magic bullets, right? Remember Paul Ehrlich's magic bullets? Antibiotics or drugs that will specifically target, say, a bacterial pathogen without harming the human host. So indeed, there's a family of antibiotics called the quinolones, and probably one of the oldest ones is nalidixic acid. And I need to check this one out. I need to see if this one maybe had some toxic properties. But the specific quinolone, I'd like you to remember, belongs to a subgroup called fluoroquinolones. And the one that, please remember for lecture exam 2, it's called ciprofloxacin or Cipro. And ciprofloxacin became famous because many, many years ago, when some person that had mental health problems was sending Bacillus anthracis endospores through the mail, so anthrax spores through the mail. And this was really dangerous because when people would open up these letters or packages containing the anthrax endospores, the endospores are so light they aerosolize, and then you inhale them. And then the endospores germinate in your lungs, and you end up with inhalation anthrax, which can be rapidly fatal, right? And initially, people might think they just have the flu, right? But if they don't get the antibiotics in time, they're going to die. So there were many people being treated with ciprofloxacin or being treated prophylactically, preventively with ciprofloxacin if it was thought that they'd been exposed to these anthrax endospores. So I think it made the cover of Time magazine at that time. So do remember that ciprofloxacin, it's going to inhibit bacterial gyrase. If we stop bacterial gyrase, then the DNA can't be relieved of supercoiling, chromosomes can't be copied, so the Bacillus anthracis or whatever bacterium you're treating can't divide. And again, indirectly, you can shut down protein synthesis. And that too is important because Bacillus anthracis makes protein toxins, right? So you stop them from dividing and you stop them from making the protein toxins. I threw these other fluoroquinolones in just for those of you that maybe are interested. Maybe some of you are going into pharmacology or pharmacy. So another one is norfloxacin, and these are some later ones, gatafloxacin, gemifloxacin, and apparently these are really good for also treating urinary tract and respiratory tract infections. Okay, this is our big overview of bacterial DNA synthesis. Again, we're going to zoom in on just one of the replication forks, but these are, you could almost practice by making these exam questions. So you guys, what's the enzyme that's going to copy the DNA and make the DNA? It's DNA polymerase. We want to remember that the direction of DNA synthesis is always 5' to 3', and we want to remember that the new DNA strand is going to be synthesized anti-parallel to the template strand. Recall we said that DNA synthesis is bidirectional, and these next tidbits, you guys, right now this won't make any sense, but we'll just describe them, and then we'll go into detail. So DNA polymerase, it can't start DNA synthesis all by itself. It has to have a short little RNA primer to get it started. We'll see that in the chromosome there'll be 2 new strands synthesized. One of the strands, we call it the leading strand, and it's synthesized using continuous DNA synthesis. The opposite strand is called the lagging strand, and it's synthesized using discontinuous DNA synthesis, which we'll describe. This lagging strand is made in a series of DNA fragments called Okazaki fragments, right? And then when we look at problems and solutions, we'll see that 2 problems that you all are going to recognize is, if we have these RNA primers in the chromosomal DNA, how do we get rid of them? So we'll see that a special DNA polymerase called DNA polymerase 3 is going to come along, remove the RNA primers, and replace them with DNA. And then the last problem that the cell will solve is these DNA fragments, Okazaki fragments. They have to be covalently linked together, and that's going to be accomplished by this cool enzyme called DNA ligase. DNA ligase is going to link all the Okazaki fragments together. So folks, I think what I'll do here is I need to learn to make these videos shorter. So let me see if I can stop the video here and get this posted, and then we'll come back and we'll use this diagram. Your diagram in your packet is much better because here they haven't used specific names of the enzymes and proteins. The diagram in our class handouts are a lot better. They have the correct names. So we'll walk through all the steps at the replication fork so that we can take a look at how the leading strand of DNA is made, how the lagging strand of DNA is made, and how the cell has evolved solutions to all the problems it's going to face. Okay, so let me see if I can't close up here, and then we'll do a DNA replication part 2 video. Okay.